today's speakers, we have two speakers hosted by GDR RNA. So the first speaker will be Jean-Louis, and the second speaker, Alexandre. And I'll come back with a brief presentation of each speaker. But before, I'll make a short introduction about the GDR RNA. One announcement also, we are just today launching the Discord platform for RNA Collaborative. So you will find the registration link on the Q&A, so you can copy and register. So we invited to join us at on the Discord platform by clicking on the link which is provided, and then eventually we can have a chat after the webinar. So first I will make a short introduction about the GDR RNA, which was created with a special topic on RNA as a tool and target for medicinal chemistry and chemical biology. It's chaired by Maria Duca from Institute of Chemistry in Nice and co-chaired by Karine Tisney in Paris. So it brings together different research groups around France, and about 300 people if we include researchers and young researchers, PhD postdocs. Most of them are also affiliated to the National Institute, CNRS, in CERN for the respective laboratories. And as you can see on the map, with many different research groups in different universities among France. Okay, so what are the goals and motivation? So it's designed to develop joint research efforts, in particular in some aspect like RNA targeting, RNA modification, and use of RNA as a tool. Also to interface the Institute laboratories working in the RNA field at the frontiers between chemistry and biology. Also, we intend to instigate a cooperation between research labs and initiate that should allow to initiate new collaboration and innovative projects. And of course, as a result, it should increase the visibility of the French community in Europe and worldwide. And one important aspect also for our young researcher is to educate them to multidisciplinarity and interdisciplinarity. So the GDR is actually organized in 3 different axes, depending on the respective expertise of the research groups, which are the member of the consortium. So the first one is focused on the design and synthesis of tools, in particular for small molecular RNA ligands and modify RNA oligonucleotides. The second one on the study of interaction, and the third one on therapeutic applications, both in vitro and in vivo models. So it goes from chemistry, in particular for the synthesis of oligonucleotide small molecules, then to the methods approaches to study the mechanisms, binding molecular interaction, then a structural biology approach to study the complexes, and activity in vitro in vivo studies. So we organize regularly events and scientific animations. So the first scientific meeting actually took place last year at the end of the year. We have a young researcher meeting coming in a few days, and the second annual meeting will take place in Bordeaux in France in the month of October. So you can find also information about the GDR on internet, Twitter, LinkedIn, and the website. We also interact with other consortium of society, in particular the GDR Chemobiology, with a large network of chemical biologists and medicinal chemists. with the CIF RNA, which is a kind of French RNA society, and the Society of Pharmaceutical Chemistry. They organized last year a symposium on drug discovery in the RNA world. And so, as I mentioned, our first speaker is Jean-Louis Mergny. So Jean-Louis is a group leader at École Polytechnique near Paris. He has developed an expertise with a variety of experimental approaches to study non-canonical DNA and RNA structures, such as G-quadruplex or iDNA. His projects encompass the structural and biophysical characterization of these motifs, the analysis of their interactions with drugs or proteins, as well as the conception of novel production tools. So, Jean-Louis, the floor is yours.
Thank you, Mr. Chairman. So it's a real pleasure to be among people working on RNA. There are friends I know among the participants, so welcome everyone. So today I will speak about quadruplexes in general and try to convince you that quadruplexes are everywhere, including on RNA. So I've been working on two types of quadruplexes, which are on the left, the G quadruplexes that you may know, and on the right, the I motif that you perhaps never heard about. And maybe the reason why is that the I motif is formed by C-rich sequences. It's based on hemiprotonated C plus base pair. Because of the pKa of that group, this base pair will be mostly stable at acidic pH. But the reason why you may perhaps don't know this structure is because the stability of the RNA I motif is much lower than the DNA I motif. And therefore, it is not very likely that the RNA I motif is physiologically relevant, although DNA I motif has been demonstrated to exist within human cells. So let's focus on what is happening on the other strands with the G-rich motif. And in that case, if you have blocks of guanine, you can form what we call a G-quartet, which is based on the pairing of 4 guanines. And if you stack 2 or more quartet, you will form what we call a G-quadruplex. In between quartet, you can put a cation, such as potassium or sodium. Potassium and sodium are very abundant in the physiological medium. So basically, you have everything you need within the cell to form stable quadruplexes, DNA or RNA, under physiological conditions. Quadruplexes have been studied for their importance in biology and in various pathology, first cancer, and then on other diseases. And I will talk in the end about viruses. There are a number of molecules that can bind to G quadruplexes. The initial goal was to inhibit telomerase. Perhaps this is not such a good idea for reasons I cannot go into details today, but there are plenty of other functions for this quadruplex ligand. Some of them can be related to RNA, and they can be used either as probes or as drugs. And regarding drugs, 2 molecules went into clinical trial as quadruplex ligand. But in addition, I would like to mention that there are other compounds known for different activities which went into clinical trials or are FDA-approved drugs and bind more or less strongly to G quadruplexes. You may have noticed that there is often a common feature for most of the G4 ligand. They are aromatic planar molecule, and this compound that we recently described is nicely fits the dimension of a G quartet, so it can stack on a G quartet, And actually, it can be sandwiched between two terminal quartet from two different quadruplexes. So this is not exactly intercalation in this case, but a sandwich between two quadruplexes. The problem we are currently facing is the specificity of these ligands. So first of all, in this test, we tested plenty of ligands against plenty of different quadruplexes, some DNA and some RNA, and two duplexes as well. This work is almost a decade old, but I don't think the new ligand would make a very strong difference if we were adding them in this table. Basically, we have solved the issue of the specificity between quadruplexes and duplex. So we have compounds that will bind to quadruplexes, but not to duplex or single strands. On the other hand, it's quite difficult to design a compound that will bind to one quadruplex, for example, in the TRF2 messenger RNA, for example, and not to other quadruplexes. And perhaps this is very difficult to achieve with the molecules that would stack on the terminal quartet, because that's a common feature between all these quadruplexes. So now if we start to compare RNA and DNA quadruplexes, here is a result which are a bit old, but still hold, about the compare stability of DNA and RNA. So we are considering here very short oligonucleotides forming tetramolecular structure. And in this case, you will notice that RNA associate faster than DNA. So K on is higher. While on the other hand, once you form your quadruplex, the RNA quadruplex is actually more stable. Its dissociation is slower, so K off is lower than for DNA. So as you can imagine, these two effects are cumulative. And if you look at thermal stability, again, RNA is more stable than DNA. So overall, even with short sequences, RNA quadruplexes can be extremely stable, if not inert, once formed. The other feature when you compare RNA and DNA is that RNA quadruplex are less polymorphic. For DNA, you've got a lot of different topologies, parallel, anti-parallel, hybrid, and so on. Whereas all RNA quadruplexes tend to be parallel, as shown here. You have 4 strands and they all run in the same orientations. And this is true both for tetramolecular structure here or an intramolecular fold. So you need what we call chain reversal loops to connect parallel strands. There are a few exceptions about the rule that RNA is more stable, and we studied under which conditions the ribose sugar could make a difference in terms of stability with a deoxyribose. Now, can we predict which sequences, DNA or RNA, would be more prone to quadruplex formation? So the first algorithm which was available is called Quadparser. And Quadparser here is basically looking for blocks of guanine, here between 3 and 5, where you need 4 if you want to form an intramolecular structure. And you will connect these 4 blocks of guanine by 3 loops, which are by default composed of 1 of 2 7 nucleotides. So if you look, for example, at the TRF2 mature messenger RNA, this is a primary sequence, and you run Quadparser, you will find one motif here, which is quite short in the 5' UTR region. So 4 blocks of guanine in reds, 3 loops, relatively short, so a very compact quadruplex. Interestingly, this quadruplex is evolutionarily conserved between different mammals. It's quite close to the AUG. And if you want to study its stability in vitro, you have to cheat, because otherwise this quadruplex will never melt. This RNA G4 is too stable. So in order to observe melting, you need to work at very low ionic strength. And interestingly, if you mutate this motif, this is what Denis and Jean-François have done, you will observe that your messenger RNA will now be more efficiently translated. So this G4 element, just upstream of the AUG, is playing a negative role on translation efficiency, which raised some question. Why would you have this evolutionary conserved region that makes your RNA less efficiently translated than others? And where is the regulation if your quadruplex is so stable? But back to predictions. When we ran Quadparser on DNA and RNA, we realized there were some shortcomings. So Quadparser was not the perfect prediction tools. There were a few false positive or false negative. Let me give you one example. And this is the reason why we embarked in designing a better prediction algorithm, a collaboration with Yves Pommier in NIH, where we were looking in quadruplex forming motif in a very GC-rich intron in the topoisomerase 1 gene. And there were 3 quadruplex predicted, and only 2 of them were confirmed to form a quadruplex. So this one is an example of a false positive. But in fact, the real issue is more with false negative. That is, sequences that do not match the Quadparser consensus. For example, this motif has a central loop of 9 nucleotides, not 9 base pairs, sorry. And its stability is still very high, and it's forming a very nice quadruplexes. And over the years, we found plenty of examples of quadruplex forming sequences that would be stable, but escape the Quadparser consensus. If you look now again at the TRF2 messenger RNA, you will see that beside the canonical quadruplex in red, you have other regions that are G-rich, and that could form in vitro quadruplexes, and we are currently demonstrating that some of them are also forming G4 structure. So we need a better prediction algorithm. So we designed this algorithm called G4 Hunter, and the basis is to look for blocks of guanine, of course, but you would still consider isolated guanine as favorable, less favorable than a pair of guanine or a triplet of guanine. And also you will favor what we call GC skewness, that is asymmetry between a G-rich strand and a C-rich strand. So you will put a negative score on all cytosine, meaning that they will play a detrimental role on the stability of your quadruplex. You want some G-rich strand or C-rich strand if you're working on DNA and look at the complementary score. So basically what you do is to re-encode your genome into numbers. So you, starting again from TRF2, you have the primary sequences, and you will re-encode it from -4 to +4, here from 0 to 8. And then you will define a window, typically 25 nucleotides, makes the average of the value you find, and run the window on your sequence. And with that, you can plot what we call the G4 Hunter score versus the position on the primary sequence. You will identify the peak, which corresponds to the canonical quadruplex, which was demonstrated earlier. And you also find a lot of non-canonical G4 motifs that significantly deviate from this 0 value, but may form quadruplexes, at least in vitro. So that was on a short motif. You can run it on a much longer sequence, such as the P73 genes, for example. And what you will discover is that overall, you have an average value close to 0. And at some position, you will significantly deviate from 0, or extremely positive value, which means G-rich motif that can form quadruplexes. And also very negative value would correspond to C-rich motif, which will, if we are talking about DNA, form a I motif, but if we are talking of RNA, probably do not form a secondary structure. So on the P53 gene, we could confirm that some of the quadruplex could be formed and could actually participate in alternative splicing events. Again, another example, the P73 genes. Again, the average is close to 0, but you start to see local anomalies, regions which can be fairly long where the score is significantly different from 0. And if you do a close-up on one of these regions, you will realize that it corresponds to a mini satellite repeats in an intron that correspond to the repetition of a 35-36 nucleotide motif, which is C-rich in that case, so could be relevant at the DNA level and form a quadruplex or multiple quadruplex in series. Now, so that was on whole genes. You can do the same on whole genomes. In that case, we plotted the density of quadruplex we can find per kilobase as compared to the threshold we choose to define what is a quadruplex motif. And something I wanted to insist on it, I'm not giving you a figure on the numbers of quadruplex you will find in the human genome, because this number will vary, will depend on the threshold you choose. So let's say with a threshold of 2, you only select extremely stable quadruplex, and you've got, let's say, 2 every 10 kilobases. On the other hand, if you choose a more traditional score of 1.5, you will find nearly 1 quadruplex every kilobase in the human genomes. The second observation is that you've got more quadruplex in the human genomes than in Drosophila, and far more than in yeast, for example. But if you look at vertebrates, there is actually nothing special with the human genome, and the highest density is actually found in dogs and pigs. So where are they located in the human genome? Often in human promoters. And you find this peak just upstream of the transcription start site. And if you look now at the fraction of human promoters that contain a quadruplex, depending on the definition you choose, for example, using a very narrow restrictive definition of a promoter, you will still find almost half of human promoters having a quadruplex with a score of 1.5, so already quite stable, and 1/4 with a score of 2, meaning extremely stable quadruplexes. So what happens if you add a quadruplex ligand and you look at the gene expression of a number of genes? So this is what we did with a G quadruplex ligand. And what we found is that we found a number of genes that can go up or down. And this is in line with the idea that our ligand is not able to distinguish between these many quadruplexes. And also, what is striking, you actually have more genes which are up-regulated than down-regulated, which kind of contradicts the idea that quadruplex and quadruplex ligand should be seen at transcription inhibitors. There are other positions in the genome where you can find your quadruplexes, and a very striking feature is the overrepresentation of quadruplex at the junction between the first exon and the first intron of genes. And you've got a strand bias, which means that it always corresponds to a G-rich RNA, so not specific enrichment in C-rich RNA at this position. And this is only true for the first exon-intron junction, as shown more recently by another team who could find that this is not the case for other type of junctions. And the same authors found a possible link between this overrepresentation and the nucleosome structure with a protein that can be removed from this thanks to this G4 RNA. So that was for human genomes. What can we say about viruses? So regarding viruses, the first virus we were interested in Bordeaux with Samir and colleagues were the HIV genome. And in this genome, we could find 11 putative quadruplex forming sequences that were conserved between different variants. And Samir was able to show that these quadruplex, many of them could be formed in vitro. And again, you've got an example of the same sequence, either as DNA or RNA, which is more stable as RNA. More recently, we've been using G4 not on a single virus, but on 11,000 different virus genomes, and look at the quadruplex density between different families of DNA or RNA viruses. As you can see here, within a single family, retroviruses, you can have a huge variation in the density. So this is the number of quadruplex you find per kilobase. But the most salient feature we observed is that if we look at the life cycle of this virus, the one that cause acute infections tend to have far less quadruplex forming motif than the one that can, for example, integrate and call persistent infections. So this is correlative and not causative, but you could suggest perhaps a possible explanation. If you are dealing with a virus that need to very quickly replicate, transcribe or reverse transcribe, remember that quadruplex are a problem. They can be very stable. So the best way to get rid of them is to is simply not to have quadruplexes. You don't have to use helicases or other stuff. So create a negative evolutionary selection against quadruplexes. On the other hand, if we are dealing with persistent viruses, then the density of quadruplex you find in the virus genome is often close to the density you will find in the host genome. So you could propose it's a kind of camouflage. But in the fact, viruses causing acute infection got less quadruplexes. So there is one virus causing an acute problem that you know about probably, which is Sarskovitu. So the day the first genome of Sarskovitu was released, we ran G4 and unfortunately, we only found one possible candidate, and it was not even a promising one. Using, I would say, a less stringent search, people found a few weak quadruplexes that are listed here. We recalculated the G4 score for this motif. They are not super promising. Perhaps a few are formed, but actually we can say that SARS-CoV-2 genome is a G4 poor virus. So are still quadruplex relevant for this virus? Maybe yes. And the reason is the virus is coding for one protein called NSP3, and this protein contains a SUD domain. So the SUD domain for SARS unique domain was identified in the SARS virus. And interestingly, there is a conservation between the SUD domain of SARS and SARS-CoV-2. And it was shown previously that these 3 amino acids, which are necessary for SARS infection, are also necessary for the SUD domain of SARS to bind to G quadruplexes. So our hypothesis was that the SARS-CoV-2 SUD domain could also bind to G quadruplexes, but that this G4 would be rather cellular RNA, not viral RNA. So Pascal Rigolet in Institut Curie builds model in which he could start from the SARS crystal structure, made the proper amino acid changes, and show that the SARS-CoV-2 SUD domain may also accommodate quadruplexes. But the best was to confirm it experimentally. In Pasteur, Marc Lavin showed that the SUD domain, we could express it in vitro, and that this SUD domain could bind in vitro to RNA quadruplexes. We actually use the TRF2 quadruplex I was showing before. If you mutate this quadruplex to prevent quadruplex formation, then the protein is no longer binding. Interestingly, if you now add a quadruplex ligand that binds to the quadruplex, it will prevent the interaction with the protein. So you can revert this interaction by adding a G quadruplex ligand. And this compound have quite decent activity in vitro with IC50 below 100 nanomolar. And interestingly, some of these compounds show also antiviral activity in an in vitro assay, in cellular assay in Institut Pasteur. So what's next? So the idea is to identify now what are the RNA G4 targets of the viral subdomain. Again, we expect messenger RNA or other RNAs from the host cell. And treat a problem I could not go into details before. In my eyes, let's consider me as a biochemist or the physical chemist of nucleic acids. This RNA quadruplex can be extremely stable. Yet, I really strongly suggest to read this very interesting review from Stefania and colleagues on RNA quadruplexes, which indicate that these quadruplex are actually dynamic structure in the cell. So there is a kind of interesting crosstalk or contradictions between these two observations. So possible explanations are maybe we should also take into account in our predictions low stability quadruplexes, and this will modify our criteria for quadruplex predictions, or also, and this is not self-excluding, that some helicase or other mechanism may really help unfold these quadruplexes in vivo, making stuff which is rock solid in vitro less so in a cellular environment. Also remember that there are other structures that could participate either at the DNA or RNA level in the stability of G quadruplexes. There are plenty of questions that remains to be answered regarding quadruplex, regarding folding. We start to understand DNA. Is RNA fundamentally different? Perhaps yes. And can we improve predictions? Can we get G4 ligand that would bind to one topology or one type of nucleic acid or another? And finally, if we look at the biology, can we say that quadruplex are only simply a nuisance or a problem, or can they play regulatory roles? And we have at least some evidence of positive roles of quadruplexes, at least at the DNA level. So I'm done. Just thank the people in École Polytechnique who work with me, but also plenty of collaboration. I mentioned you've seen names on the slides, on some of the previous slides, either in France and also in the Czech Republic and in China. I'm almost done. I would just to advertise the G4 Society for G quadruplex aficionados like me. We have a series of seminars on quadruplex every Thursday. I'll speak tomorrow, but on a completely different topic. And I would like to thank you for your attention. Just as a final comment, today is the first day bars are going to open in France, and I promise my wife I will only have one beer, and I will stick to my promise. Thank you for your attention.
Thank you, Jean-Louis, for the presentation. So we have a few questions. There were some general questions and some more specific questions. So maybe I'll start with the more general question, although you partially answered to that, but it was a comment from Scott, remembering some experiments from Bartel's lab at MIT. Sure. Where they were probing the G4 structures and thought that they were basically unwound in vivo. So I don't know whether you want to give more details about that.
So I can comment this science paper, 2016, I guess, which use DMS in vivo. And basically, the conclusions is that you find many quadruplexes form in vitro that are unfolded in the cellular environment, which is not that different from what I'm saying now is that in my hands, these RNA quadruplexes are super stable. We're talking about lifetimes or of years, for example. And whereas if you speak with RNA biologists, this quadruplex would be more transient. Actually, Bartel used relatively long DMS treatment time. So if the RNA quadruplex is dynamic, you will not see it. And the second comment I would like to make is that the way we define the quadruplex, it's based on the sensitivity with or without potassium. They are actually missing, by this definition, the most stable RNA quadruplexes because they would be already formed in their conditions without potassium. So we checked some of their sequence, and some of these quadruplex are not reported as forming a quadruplex simply because you only need traces amount of potassium to form. And finally, the idea is that this is true for eukaryotes, and in prokaryotes, you don't have such unfolding in the cellular conditions, but you've got a much stronger evolutionary pressure to prevent the existence of stable quadruplex. Because let's say I'm oversimplifying, but the bacteria cannot deal with them as efficiently as would a eukaryotic cell. So the best way to deal with them is to get rid of them. That was a little bit what I was saying about viruses.
Okay, so we have two more specific questions about TRF2. You mentioned as a model for the messenger RNA. So the first one is TRF2 messenger RNA more stable because of the G quadruplex in the 5' UTR.
Oh,
and yeah, maybe I'll go with the second one, which is related. Does G quadruplex in the 5' UTR or 3' UTR impact stability? And does it always translation? Like
so I will start
TRF2?
with the second questions. Okay. And the answer is no, it does not always hurt translation. And there have been examples in which quadruplexes in the 5' UTR could play a positive role on translation in the case of cap-independent translation. So this is not always detrimental, and it will also depend on the position of the quadruplex in the 5' UTR. About the first question on the lifetime of the RNA quadruplex, my answer is I don't think so. I'm not entirely sure. Denis Gomez could probably answer that one. But just say that to mutate this quadruplex, you only need to change a few nucleotides, and that will be enough to kill quadruplex propensity. My expectation is no, but I'm not 100% sure.
Okay, so there's still a few questions.
All right, there is a question from Clément. And that's about 2' of methyl and RNA G quadruplex stability. We have studied some time ago various nucleotide DNA RNA analogs for quadruplex propensity. Many analogs are compatible with quadruplex formations, including 2' of methyl. And what we found is the 2' of methyl tend to have an intermediate stability between DNA and RNA. We don't have a high resolution structure of a 2' of methyl RNA G4. So I cannot really explain why the stability is intermediate. But many analogs got higher stabilities than DNA, for sure.
Okay, so Pedro has questions about the rational design efforts for G4 binding compounds.
All right, so
are the existing ones selected from a chemical library with a lot of diversity?
So let's say it's relatively easy to design a quadruplex ligand that will bind to quadruplexes and nothing else. That is achievable. What is much more challenging is to design a ligand that would bind to your quadruplex of interest for example, the TRF2 short quadruplex I was describing, and not to another quadruplex. You have some ligands which show preference for some topologies. For example, they prefer parallel quadruplex over hybrid or anti-parallel. That is doable. But if you were to, for example, design a compound that would bind rather to a RNA parallel quadruplex and to a DNA parallel quadruplex, then clearly stacking on a terminal quartet is not going to be sufficient to achieve this selectivity. You will have to play with the nature of the groove. Only one is available because of the chain reversal loop, and it will be a very challenging task. So in principle, you could. In practice, it's probably extremely hard.
Okay, there was a short question before. How does G4 enter or quad parser perform compared to QGRS?
All right, so QGRS depends on the threshold you choose. The problem with anyone looking for G quadruplex in a genome is that when you start not to find some, you can always lower the stringency until you find candidates. This is true for QGRS, it's true for G4 Hunter. But then you will find a lot of false positive or very low stability quadruplexes. In our hands, the point with QGRS, it is generating a score, but this score, let's say, was not directly correlated with experimental data. In our hands, to validate G4 Hunter, we worked on hundreds of different quadruplexes. I must confess, mostly DNA, because of costs. We had less RNA, so we knew that it could be more accurate. We are not yet far from 100% accuracy, but we have an accuracy which is reasonable.
Okay, we'll take the last question from Vasiliki. Do you have an estimation of how many RNAs can form RNA G quadruplex? For example, you said one or four of promoters could form G4s. What about RNAs and maybe more specifically non-coding species? Can small RNAs also form RNA G quadruplex?
Well, I'll start by answering the last question, and it's clearly yes. For example, tRNA or tRNA fragment have been reported to form quadruplexes. In addition, for example, the RNA component of telomerase HTR or HTERC, its 5' end can form a stable quadruplexes. So these are two examples. About the number of quadruplex you may find in RNAs, my honest answer would be, I have no idea. And the reason is, I can always choose the threshold for the search that will give you a given number. The problem will be what will happen in vitro. Maybe I could tell you, if I were to fold my RNA in vitro, how many would I find? What will happen in the cell is a different question. And also, I need first to answer that question, to answer the question I mentioned before, is that are two quartet RNA quadruplex relevant in the cell? If yes, this one could be far more dynamic, ready to open than the others, and they could play regulatory roles. So order of magnitude, 10 of thousands maybe, but I cannot be any more precise than that.
Okay, thanks a lot, Jean-Louis.
Okay,
so
I'll
we'll
stop
now,
sharing.
yeah, thanks. Now switch to the other.
Sorry, I did the wrong way. Excuse
okay,
me.
no problem. Yeah, I'll put back just to present Alexandre, who's the second speaker of the day. So Alexandre is a group leader at the Institute of Functional Genomics in Montpellier, cell biologics expert in translation compartmentalization. He has contributed to major achievements in the field, in particular on viral-induced tRNA methacylation discovery, nuclear translation visualization, MSc recruitment in specialized translation sites, and invention of ribopuromethylation method. His current research focuses on the contribution of post-transcriptional mechanisms on cancer cell adaptation, in particular RNA epigenetic and translational control. Within the framework of SMART Consortium and the RNA GDR, is implementing unique tools and techniques at the crossroad of basic, applied, and clinical science. So, Alexandre, the floor is yours. So you can start sharing your slides.
So first, thank you for this nice introduction. Hi, everyone. So do you see my presentation? It works?
Yes.
Good. Okay. So first of all, hi, everyone. It's a pleasure to have the opportunity to present this work. So as Fabrice was mentioning, I'm really interested in translational control, and lately we were more interested in epitranscriptomic and how it can affect translation. So we work on cancer, and colorectal cancer in particular, and we are very interested in one specific characteristic of cancer cell, which is adaptation. And I really like this quote from Charles Darwin, saying that basically in cancer, in the example of cancer, it's not a matter of strength. You know, it's not the cell that proliferates the most or like the strongest cell that will survive.
Alexandre, we may have some trouble with the sound of your microphone.
Sound?
Yeah.
Okay. Is it better?
Yeah, I don't know. I have some kind of distortion of the sound.
Same for me.
Okay. Thank you, everyone. Okay, sorry. Just give me a moment.
Do you think it's a network problem of the bandwidth? Maybe you may try to turn off the camera, see if it's better. Yeah, you may stop the video and.
Does it work better now?
Yes.
Okay. Yes. So it depends. Sometimes it stops working. Okay. So it's better. Yeah. I would share the screen again.
Sure.
So I think. Yeah. Right. Yeah. So now it should work.
Yeah, and it's okay with the sound.
Okay, good. Okay, so maybe you listen to that part. So yeah, we are interested in cell adaptation, and in particular how a cancer cell can adapt to different situations. And finally, the cancer cell that will survive is not the one that is capable of proliferating the most, but the one that is able to adapt. And the striking illustration of these features in cancer and in colorectal cancer in particular is the cancer stem cell. So cancer stem cells represent a small subpopulation of cells that are present in cancer, in the tumor. And these cells they have an annoying ability. In particular, they can self-renew, and they can also resist to therapy, and they can initiate tumor in distant organ. And this is what is striking about this cell. This cell can adapt to different kind of stress and environment. So they can leave the tumor at stage 4, they can be found in the circulating blood, so they can adapt to a completely different environment. And finally, they can enter a new site, which is completely different from the primary one, and they can initiate a metastasis in this new site. So they can really adapt to different kind of stress and environments they are present in. And we are really interested in that, how these cancer stem cells have this amazing ability and how they can also differentiate or de-differentiate depending on the environment, depending on the cellular context. So to be able to adapt, you need to be fast, because if you need hours for complete reprogramming of the cell, then the cell won't survive. So at the beginning, we were really interested in how translational control could quickly allow the cell to adapt to different stress. And more lately, we've been really interested in epitranscriptomic, which are modification of RNA. And now, basically, we believe that these modification and their dynamic can be very useful for translational control and to provide this adaptation capability. So we started working on the modification everyone works on, which is the N-methyladenosine, M6A. And 2 people of the lab have been working on that project, Sebastian and Amandine. And just to give you a brief introduction about M6A. So M6A is present in different types of RNA, and in messenger RNA in particular, it is present in close to the stop codon and in the 3' mutation, sorry. And so like you can see, you can find many others, up to 14 different modifications in this messenger RNA, but most of them actually have specific location, which is quite interesting. And M6A, when we started the story, and I will try to tell the story the way actually it happened. So at the beginning, we did some research on what was published on M6A, and M6A, if we look at biological process, was involved in many different ones, and some processes were more interesting for us, such like stemness and differentiation, pluripotency, self-renewal, all these characteristics that are really related to cancer stem cells. And in the pathology, when we started the project, many papers were already out about how M6A was involved in cancer in different ways. But there was nothing on colorectal cancer. So we started with a very simple idea. So maybe M6A is involved in cancer stem cell adaptation and in cancer stem cell abilities in general, which means that one of the actor of M6A, one of the effector, is probably capable of balancing, like this switch from cancer stem cells to non-cancer stem cells. So you can find different actors involved in M6A dynamics. So you have a writer complex, which will add M6A. You have different readers. So when we started actually the study, there was only YTHDF1 and 2 that were published. And you have 2 known erasers, which are LKBH5 and FTO. And we decided to start with a very simple experiment to test functionally whether one of these actors could be involved in cancer stem cell ability. So we used a cell formation assay. So cell formation assay is basically to put the cells, so you put the cell in suspension culture at very low density without serum. And the cells that have some stem-like abilities will be able to form spheres 7 days later. And these are actually spheres you can see after 7 days. And we transfected siRNA targeting individual effector of M6A, and we tested whether inactivating individually one of them could impact this sphere formation assay. So this is just a protein level to show you that all the actors we targeted, all of them were almost completely silenced. And when we look at sphere formation assay, so this is the control. So in general, you have like between in small well we have between 10 to 15 spheres, and for most of the effector, like the writers, the readers, and LKBH5, we had no effect on sphere formation assay. But for one of them, which is FTO, we had an effect with 2 different siRNA. And this effect, so you silence FTO and you improve the ability to form sphere. And this effect was actually observed in 6 different cell lines. So that was not depending on the type of cell line you use. And we used cell lines derived from patient, but also common cell line, such as SW620 and HCT116 here. So that was very interesting. And we were surprised to see that only one of them actually did something. So we designed stable cell line, 2 different stable cell lines. So SH control cell line, which is basically a cell line expressing SH, a random SH, which is supposed not to target anything, and SH-FTO cell line, which is actually down-regulating FTO expression. What is important is in these 2 cell lines, when you compare the FTO expression, in SH-FTO, you have only 50% decrease of FTO expression, that's a very important parameter, because we tried many different tissue and cell line, and you don't have like a 10-fold decrease or increase of FTO when you compare the cells. So there is no such thing as a FTO knockout, and that was a really important thing we wanted to do. So these cells were injected in mice, so different number of cells, 1,000, 500, and 100 cells. So they were injected subcutaneously, and then we waited almost 50 days to look at the tumor. And surprisingly, 50 days later, so these are the control cells. So you have some tumors, but not that many. Even with 1,000 cells, only 2 mice out of 5 were showing a tumor. But in the SH-FTO, you have a tumor almost all the time, even with 100 cells only. And that was quite surprising, because just a decrease of 50% of FTO will be enough to increase 8-fold the ability to initiate tumor in mice. And then we look at the ability of this tumor to resist chemotherapy. So we took the exact same cells, but we used a little bit more to make the tumor bigger. We waited until we achieve a certain mass of tumor, and then we treated the tumor for 60 days with 5-FU plus irinotecan, which is the regular therapy for colorectal cancer. And surprisingly, while the control mice with SH control cells actually show a stabilization of the tumor mass, in the SH-FTO mice, the cells didn't care. Basically, they kept growing. So the tumor was insensitive to therapy. So not only with 50% less FTO, we had more tumor, but these tumors were insensitive to regular chemo. That was the first, basically the first conclusion we drew. So when you decrease, not abolish, but just decrease by 50% FTO expression, you push the cell phenotype toward cancer cell phenotype. And I didn't have time to put that in this presentation, but you also have an increase of biomarkers that are specific for cancer stem cells. Okay, so next question was, does it happen in real life? Because like all of this was in vitro and with cell lines. So we use different cell lines in the lab that are isolated from different tumor, from different patients. So primary tumor, cell lines derived from circulating tumor cells, but from blood, and cell line derived from metastatic sites. And these patient-derived cell lines were actually analyzed for FTO expression. And what we saw that actually the ones with the less FTO expression were the CTCs, so the circulating tumor cells. And that was quite striking compared to the primary cells and metastatic. And actually, our lab have shown before that these CTCs are the cell lines and the cells in general that have the most cancer stem cells hallmark if you compare to other cell lines. So actually, they have a kind of inherent ability to resist therapy and to form tumor in mice. So the next question we ask, because basically what we see is in circulating tumor cell, you have less FTO. What if you increase this expression? Because this cell line, this CTC cell line, they are more stem-like. If you increase FTO expression, can you revert this phenotype? So that's what we did. And actually, this is true. When you increase FTO expression by overexpressing FTO in the CTC cell line, you decrease the sphere formation ability, and you decrease by half their ability to resist therapy. So it goes in both directions. So now, when we had all this data, we thought, okay, so what's happening? Why there is this change of phenotype? What is happening at the molecular level? So FTO was known, of course, as the M6A demethylase, and at the time there was a big fight because, like most of the publication were showing that FTO was actually targeting M6A located at the 3' UTR. So this is that was basically the target. But one paper came out and others showing that actually FTO can also demethylate this site, which is the plus one. It's the first adenosine adjacent to the cap. And this adenosine is already methylated here. And this in this paper from Jeffrey's lab, actually, they show that that was the favorite substrate of FTO, but only in the cytoplasm, not in the nucleus, because in the nucleus that M6AM was unavailable, that was already occupied. And at the time, a lot of things were known about M6A and its role in different biological function, but there was nothing really on M6AM, and there was always this big fight about what's really happening, what is the real substrate, and we didn't care. I mean, we were just interested in the truth. So we didn't have anything when we started this this project to measure methylation on RNA. So we had to build the consortium in Montpellier and develop technology, develop mass spectrometry and bioinformatic pipeline to analyze epitranscriptomic and design new tools. So this is basically the SMART consortium. So this is what we have built in Montpellier, and thanks to that, we've been able to develop, I mean, many projects and answer the question we had for this one. So the workflow we use for analyzing epitranscriptomic is first to, of course, pick the sample and isolate RNA. So you can isolate different species of RNA depending on the kit or the protocol you use. And from this RNA isolation, then you process RNA, you cut down the RNA, you get nucleosides, and we can analyze simultaneously more than 40 RNA modification by mass spectrometry right now. And thanks to that, we've been capable of answering the question, so is it M6A, M6AM, or both? And first, we look at M6A level in SHFTO cells. I showed you that the phenotype was quite strong, and there was nothing. So we were quite surprised to see nothing. Then we look at M6AM, and M6AM actually was twofold, showed a twofold increase following FTO shutdown, which was going in the right direction because you remove the methylase, so you get more. So that was quite surprising because it was not what we were expecting. So we look at all the cell lines we had. Remember, like the cell lines from primary tumor, from circulating tumor cells, and metastatic. And M6A is quite, it's quite not the same depending on the cell line. The level can change up to twofold, but there is no a clear consensus. But when we look at M6AM, we can see this of M6AM in CTC cell line, which was concurring with a decrease of FTO level. So that was going in the right direction. Like everything was taking shape, I would say. And at this time, approximately, this science came out, and PCIF1, which is the methylase of M6AM, it was discovered. So that was the right timing, I would say. So we thought, okay, so now we can see whether we could do the opposite, targeting the methylase this time. So that's what we did. We targeted PCIF1, and we could decrease by 30 to 40% the ability to form sphere in culture. So that was the opposite of what we had with FTO. So it was going in the right direction again, and we decided to perform the ultimate, basically the ultimate experiment that will prove that this is the right mechanism. And we targeted PCIF1 in the cells where FTO was decreased to see whether we could balance the effect. And that's what you see here. So this is the SH control cell. These are the SH-FTO cells. So you increase by twofold the sphere forming ability, and then you decrease PCIF1 also, and it goes back basically to the SH control cell line. So you could cancel the effect basically. The only problem was that what was published about M6AM at this time was that M6AM was demethylated by FTO in the cytoplasm, and FTO was supposed to be nuclear. So we look in the cell line in CRC1, which is a patient-derived cell line, and SW620, which is a regular cell line used worldwide. And in both cell line, actually, you could see that FTO is present in nucleus, in nuclear speckle, so it's quite clear. But it's also present in the cytoplasm. So in cancer cell line derived from colon, FTO is present both in the nucleus and the cytoplasm. So, and if it's present in the cytoplasm, it means it can target potentially M6AM. So we fractionated the cell, nucleus on one side, cytoplasm on the other side. We look at M6A by mass spectrometry, and we didn't see any big change. And then we did the same with M6AM, and we could see that actually M6AM was clearly increased in the cytoplasm, but not really in the nucleus. So with that experiment, we confirmed that FTO was capable of demethylating M6AM, and this enzymatic reaction was taking place in the cytoplasm and not in the nucleus. Exactly the same thing that was published before. But we have this question, why in HEK cells, for instance, you have FTO only in the nucleus, not in the cytoplasm at all? And in many other cancer cell lines, FTO is present in the cytoplasm. And we did some IHC staining on a tumor microarrays at different stage of colorectal cancer evolution, from stage 0 to stage 4. And if you look carefully, you can see that FTO by IHC is present only in the nucleus of cells at stage 0. So, so there is no tumor. But once the tumor is born, basically, so once the tumorigenic process has started, it goes into the cytoplasm. And that's what we could see in the cohort we use. So we don't see any change or clear change of FTO expression, but we cannot see it actually, because if it's only a twofold change, IHC cannot allow you to see that. But in any case, FTO, which is nuclear in the healthy tissue, following transformation of the tissue, goes from the nucleus to the cytoplasm. So it becomes cytoplasmic following a tumorigenic process. And basically, so this is what we are, and this is where we are right now. So we are trying many things to now understand what is going on. Okay, so what we know is that M6AM, with this enzymatic reaction, with enzymatic equilibrium with PCIF1 FTO, so this modification can actually balance between a cancer stem cell phenotype and a non-cancer stem cell phenotype. So there is a clear effect of this modification on this phenotype. But what's really happening? So we are trying now to see whether M6AM could affect the cap structure, and it would change translation. So what we have been doing until now. So we did some sequencing, of course, already, but at steady state, we cannot see any change. So that was surprising. So M6AM, when we look at SHFTO and SH control cells, I've shown you before. So you see that there is a huge, dramatic change in phenotype. But if you look at the transcriptome and the translatome, you see nothing. I mean, there is no change. And we look very carefully. So what we think is that M6AM will change the structure of the cap, and that will be useful under stress condition. So this is what we are doing right now. We are trying to understand how M6AM could help resist stress and could help the cell, the cancer stem cell, actually to adapt to the environment. And the other question, so this is the last thing I show you. The other question we are trying to answer right now is why FTO relocates from the nucleus to the cytoplasm following transformation. And that is something that needs to be understood, because this way, all the people that will be, fighting about these differences between HEK cells and other cell lines, and what is the substrate of FTO, everything could be explained. Why FTO is changing location. So that's another question we are trying in my group to answer right now. And I would like to finish on that and take any question. And thanks all the people involved in this story. So the people from the group, the people from the SMART consortium that helped develop all the tools we used, from the ICM, Maria, and people from other part than Montpellier. So Yuri in Université de Rennes and Francesca in UMEA University. Of course, I would like to thank all the people that gave us a lot of funding to make this story happen. And I will thank you for your attention and take any question right now. Thank you.
Thanks, Alexandre. So we have one questions. Assuming from Aslian, assuming you analyze total RNA M6A levels by LCMS in SHFTO cells, do you think total RNA might mask of observing M6A level changes in messenger RNA?
So we actually isolated all the species individually. Messenger RNA, tRNA, microRNA, ribosomal RNA. We tried everything, and there is no change. And we did merit sequencing too, and there was no change. The only thing changing is M6AM. So that was we were really careful with that because it was a sensitive point.
Okay, so for the moment we don't have any more questions. So I will ask one. So you checked the level of expression of FTO. So what about PCIF1?
So PCIF1 is changing too. So we can see changes depending on the cell lines. But following chemoresistance acquisition, because we made many other experiments. So actually, we made some cells become chemoresistant, and this cell, they have the same level of PCIF1. There is no change, but FTO decrease and M6AM increase. So it seems like PCIF1 is not changing a lot between cell lines from one cell line to another, but FTO show really important changes.
So you mean in CSC or no CSC, you should have the two activities balanced between FTO and PCIF1?
Yes, okay. Not completely balanced, but PCIF1. I mean, if you FTO I don't think FTO will have the same targets than PCIF1 because they are not in the same compartment. I mean, I don't think, and the PCIF1 will methylate everything, and FTO will only demethylate specific messenger RNA. That's what I think. So they won't really balance each other. So they are not completely antagonists, I would say.
Okay.
It's just if you have no methylation, FTO cannot demethylate, so it will negate, I would say, the phenotype. It's complicated.
Yeah, so you quickly mentioned the issue of localization. So can you say a bit more about what's the impact of localization? And so you would have some changes in the localization, but it would be induced by what?
So actually, so few papers suggest that post-translational modification could impact FTO location. It could be, for instance, phosphorylated, and it will stay in the cytoplasm, things like that. But it's not really clear. There is no clear evidence. There is no really thorough study explaining why FTO could be in one location and not the other. So there is a nuclear location signal. So there is a, so it should be in the nucleus. In HEK cells, it's exclusively in the nucleus, but in cancer cell, it's different. And I don't think we know why, and I don't think we know what happened.
Okay, so we have one more question from Jan. Did you evaluate any small molecule inhibiting M6A RNA demethylation?
Evaluate? No, yes, we evaluated one at that time, which was the RHINE, and I didn't put it in the paper because it worked, but it was kind of toxic, so it worked, but I don't think for the right reason. So yeah, so I didn't I didn't try I didn't put it in the story or in the paper because of that, but it worked. It was supposed to inhibit FTO, not M6A, FTO, but yeah, I mean the inhibitors right now, FTO inhibitors are much better, but we didn't have them.
Okay, just one small questions, clarification about the method used for sequencing.
For sequencing, what do you mean?
Yeah,
like it was RNA sequencing.
Yeah, but I mean, probably was asking whether you did some standard RNA sequencing, or whether maybe you used some more other techniques, and particular for the.
So basically, what we looked, or what we did, we did a transcriptome like by RNA sequencing, a regular one, and we did the same thing with messenger RNA associated with ribosome, just to see whether mRNA engagement by ribosome could be affected by M6A in steady state. So there was no stress, and we didn't see any change doing that. So it was like our SH control cells and SH-FTO cells were completely alike. But one was really bad, like more aggressive than the other for some reason.
Okay, so we'll take a last question from Sergey. If I am not mistaken, nuclear FTO is granulated. Did you check with what it is co-localized?
No, but that is known. It is known that FTO goes to nuclear speckles, but I didn't work on that part. But it is you can find paper on that, so it's not something new. But yes, it's in specific location in nucleus.
Okay, thanks, Alexandre. So we are a bit over time, but I will just take one more minute to make a few announcements. In particular for the next meeting. Sorry. So the next webinar from RNA Collaborative will be hosted by our colleagues from EMBL. So you have the names of the two speakers for June 2nd. So in 2 weeks. I wanted to make a quick announcement about events in May until the end of the month. So from our colleagues from EMBL in Italy, A Brave New RNA World, which is a virtual event. Of course, probably many of you are already registered for the RNA meeting, which is also a virtual meeting. And also a meeting tomorrow from the University of Rochester on RNA structure and function cluster meeting. And I would end reminding you that we have this nice platform, Discord, you can join, which is intended to facilitate the exchange either after the webinars or whenever you want. So we invite you to join the space, RNA collaborative space on the Discord platform. So the link was given in the chat, and you can always contact Martina or anybody from RNA collaborative to have the details about the registration. So hopefully looking forward seeing you to the Discord platforms. Thanks for attending.